gardens, but maybe I can assume that you can easily mix with anyone in Katanga and sit there freely without hesitation. That is homogeneous mixing. Random mixing is randomly you can maybe move from one place to here. Maybe, let me say, I can move from here to Gulu, and I can easily mingle with anyone. Randomly, I can easily randomly, or I can easily meet somebody and the process continues. So homogeneous, we assume we are all equal. We can easily interact with anyone. And it is similar to random mixing. Random is that I can go anywhere at any time and start whatever I'm doing. So as you write these models, you can easily, you can make some assumptions that can guide you in, for example, making informed decisions. Because when you assume that the population is mixing randomly, or when you assume homogeneous mixing, then definitely you're going to make some underlying assumptions why you think I can easily move from here. I can easily move from here and go somewhere else. Or I can easily have equal chances as someone else, for example, in Gulu. So how exactly are you going to make these assumptions? But you also need that, as we move on, you will say that some of the assumptions we make might not really give us the best. And we are going to see how or why in all the models you've talked about that you looked at yesterday. Why are they SAIR? Why not SIR? Why not SIRS? Why SIR? All those are underlying assumptions. Then when you're looking at population heterogeneities, that is, if it is not homogeneous, then you're considering heterogeneities. In real population, almost everything is heterogeneous. That is, not two individuals are completely alike. But in homogeneous mixing, we assume people are completely alike. So then you ask, if you're going to look at heterogeneities, the important question is, do different heterogeneities affect epidemiological rates of mixing? Can parameters be estimated to describe their effect? So the question is, when do you use homogeneous? When do you use heterogeneous for populations? By just looking at that image, you see people are different in terms of the work we do, in appearance, in structure. So at times we tend to gravitate to the people we think we identify with or we think we align with. But when you're modeling a situation, you cannot easily know who is who when you're looking at population. That's why it is important to decide which approach you're using. Then you ask yourself different questions, and then you make aligning assumptions that can help you get meaningful output. So for such situations, we always use multi-group models. That's when you separate depending on characteristic or networks or individual-based models. But these are partial differential equations, but I don't think you've done those before. So why do we make assumptions? Assumptions are necessary to solve open problems because important aspects of the problem situation are not specified and additional information is needed. So the assumptions, the important aspects of the problem are not specified and additional information is needed. So assumptions specify the missing information and help the problem solver find a solution under the restrictive conditions that come along with making assumptions. So assumptions specify the missing information and help the problem solver find a solution under the restrictive conditions that come along with making assumptions. And why are assumptions important? They guide argumentation, evidence generation, and conclusions. One reason assumptions are important is that they frame and guide, directly or indirectly, then argumentation, evidence generation, and conclusions. So assumptions are the foci for any theory and thus a paradigm. It is important to make assumptions explicit and to make a sufficient number of assumptions to describe the phenomena at hand. Explication of assumptions is even more crucial in research methods used to test the theories. So the power of an experiment is only as strong as the clarity of the basic assumptions which underlie it. The power of an experiment, in this case, the power of the model that you have, is as strong as clarity of the basic assumptions which underlie that model. You have to make assumptions that are easily understandable, that make biological meaning, not just an assumption to simplify your mathematics, because most of them do, but you don't just make any assumption such that you get that you become lazy with what you're doing. So they not only underlie laboratory experimentation, but social evaluation research as well. In all labs, in all social research, there are some assumptions that are being made, because you cannot have a homogeneous environment. You cannot conclude that everyone in this population is mixing randomly, because we don't. We choose who we mix with, but we always assume we do. Assumptions can be related on a scale of articulation from tacit to explicit. So these are the models that you told me you went through yesterday. And then I don't know if you went through the reasons why in some cases you have SEI or SIR or SI. So we are going to go through the assumptions in these basic models. Now, when you look at the first model, you have SEI. I know you know what S stands for, right? You know what S is?
Yes, we know.
Yes, we do.
And you know what E is and what I is?
Yes.
Yes.
Oh, and why do you have class E and why do you have class I? Maybe we can, in making assumptions for these basic models, we can make it interactive such that you can explain why in some cases you have E, in other cases you don't have E. Can you maybe explain why you think that in this first case you have SEI, and you see we are stopping at I. Here we are going all the way to R. Here we come back to S. Can we discuss the underlying assumption, the underlying facts or assumptions we are making here?
Doctor, can I try?
Yes.
Yes, so I think for the case of E, we are assuming that once a person is susceptible at S, they can come into a compartment where they're infected, and then they pass on infectious, but then in the second scenario, it's infectious compartment, which means the moment they get maybe the disease, they're straight away infectious. But then for the infected, they may okay.
Dennis, your network.
Is it my network?
I know it's, I think, Dennis.
We missed some of the explanation. Anyone who wants to supplement that?
Try. I hope you can hear me. So for the first diagram, so one moves from the susceptible group to exposed. So it means maybe there's a latent period as the virus or the parasite or the microorganism really, the load in the system increases so that they become infective. But for the second diagram, where someone transitions from being susceptible to infectious without going through the latent period or the asymptom or the incubation period, really. So once you get the virus or the parasite, for example, you immediately become infectious and you can be able to infect other people who are, for example, susceptible. And of course, thereafter, once someone is infected, after interventions, for example, prophylactic interventions, they are able to recover and they clear out the virus or the parasite. Yeah, so that's the difference. Thank you.
Okay, thank you very much. So you brought out the reason why here we have an E and here we don't. And the reason is that it's assumed that some diseases, or it's the fact that some diseases take long to manifest inside the system. For example, HIV takes like 6 weeks before it can be detected in the system. Malaria takes 2 days, 2 to 3 days. But then there are diseases that once immediately when you come in contact with somebody, you can just show the symptoms. Some diseases you take long to show the symptoms. That is the latent period, the duration. So when you assume that this time between you being exposed to you showing clinical symptoms, you infected, but you're not showing symptoms, then you add the E. So the assumptions we normally make here is some people even in this state can infect, others don't. But if we look at HIV again, an individual can transmit malaria in the first 6 weeks after exposure. Sorry, they can transmit HIV. They're not showing symptoms, but they can transmit HIV. And you've seen people with HIV, or people showing symptoms after a while and they didn't know they had it. But it doesn't mean they got HIV immediately or like the day before. It was some time back and they have continuously transmitted. Sorry, they continuously transmitted the infection. Now, when you look at this one stops at I. Again, this is a very good example of HIV. Here we know HIV does not cure. Once you have it for life. Although so this is a good example of diseases. So as you explain why you're having E, then you explain why you're stopping at I, and these are usually in form of assumptions. Say because once you get HIV, you cannot recover. You remain infected for life. Then when we come to this, we have SIR. Or if I can go back here, if the exposure period, in some cases, as you're exposed, you may not transmit. So you have to make an assumption that only the eye will transmit. Not necessarily for HIV, but for other diseases with slightly long exposure period. For example, malaria, you can see it is 2 to 3 days. You can ignore the E. You know, it is not instant. So you can make an assumption that because you want to study this, we can ignore the exposed period. Then you can remove the E for malaria. So those are some of the assumptions we make to simplify, because the focus is not on the effects of exposure. So you can comfortably ignore the E in such diseases when the exposure period is short. And that was one of the assumptions you have to make. Now, when you come here, this means that the exposure period was either assumed not to be important in the model or it is too short. And those are assumptions that you'll have to make. But then you realize that this person moves from R back to S. So the assumption here, the underlying assumptions, and it is always hard to know the assumptions that were made, but because you know what happens in the model, in the characterization, you can always know the assumption that was made. When you see this arrow coming from R back to S, the assumption is that this disease confers temporary immunity, or temporary immunity such that, or partial immunity such that after the end of that partial immunity, let's say it gives you temporary immunity for 6 months. After 6 months, then somebody's going to go back to S and the process will continue. So when you look at this, you can see assumptions that are being made. You have no exposure, so the latent period is short. There is no death due to the disease because you only see recovery going to R. You don't see an arrow coming out. Then you have this going back. Then there is temporary immunity. That means this person loses immunity after a while. Then we come to this SIS. Again, why don't we have R at this side? Why immediately after you recover, you come back to S? The underlying assumption here is that immediately upon recovery, you can get the disease again. And this is a good example for sexually transmitted infections. You can get treatment and you clear maybe in the morning, then in the evening, you can get another, all the same. So when you see such, definitely you can write down some assumptions that were made. So every time you look at a model, you should always ask yourself the assumptions that were made. It will help you understand the model easily. So here, again, there is no partial or temporary immunity to this disease. There is no death from the disease because you see all the people coming here. If there were death, you would have another arrow that leaves I not going anywhere. That means people are living through death. Then immediately upon recovery, these people can have the infection again, and you see the continuous cycle. So in all, you need to adapt a model framework to the disease biology and to your problem. What are the objectives that you're making? What do you need to accomplish with your model? And how does it align with the disease framework or the disease biology? So whatever you do, you have to look at the biology of the disease and then your objective. And there is no need to restrict to SEIR categories if biology suggests otherwise. For example, we can have, you can add a C here. C, these are usually carriers. These are carriers, and this model can also work for HIV, but you can call R the removed. Removed means people are dead or people no longer transmit the infection. They are maybe quarantined, anything that removes somebody from the scene of infection where they can no longer transmit. So when you look at this, you have movement from SIR, but then this I can either go to R or to C. So if you call C the carriers, usually C represents carriers in the model. That means some of the infected people can carry on with the disease. They are not showing any symptom, but eventually they can be removed due to the disease. So this can also work for HIV. When you write your assumptions right, you're calling R the removed. That means they are dead or now they have AIDS, because we assume when somebody has AIDS, everyone can see they have AIDS. No one can interact with them sexually to transmit further. So if you just redefine these terms, you can have another model for HIV. So these people here are the people with HIV that have had it for a long time and are in remission. And then you justify what is happening. You can even assume, you can say, okay, these people can also die of the disease. You can add death, you can add death due to HIV. But you notice that in all these models, we have not included the natural death and natural birth. As HIV goes through the populations, people continue to give birth. People continue to die. So depending on the time scale of disease process and your questions and the host demographic processes, you can add natural birth and death. So as HIV or as malaria is in the population, people are continuously giving birth. So you include death in the different compartments, as you also include the disease dynamics. So we have SIR using the infection, that is the root, but these people can also die of other causes. And you can add death on all this, you can also add birth. The assumption here now is that whoever is born is susceptible, because you see birth is only going to S. So you should be able to look at the model. This is a compartmentalized diagram, or look at the diagram, or what we call the flux diagram for this, for your model. You should be able to tell the assumptions that are being made. For example, here, everybody who is born is coming to S. That means all the babies don't have the infection that you're modeling. Then you have the natural death. Natural death, whether you have HIV or not, or whether you have malaria or not, we assume, I don't know what is the current life expectancy for Ugandans. I think it is 62, maybe. So we assume at 62, you have to die. If you got malaria today, 62 comes tomorrow, you die the following day due to natural death. So those are the assumptions that you need to know or to see just by looking at a compartmentalized diagram. And you can easily change this, and you're going to do that during the time we have in between. You can just look at one of these models, you can take a screenshot, just add natural death, add other things as you have made your assumptions, do anything. You can even add exposure here, you can add a career, you can make this model so nice. So just adding, you write down your assumptions and redefine some of these models. And we also have cases where we have diseases in the environment. When you have environmental disease, I know you know examples of environmental diseases. For example, cholera. What are the others? Cholera, diarrhea, hepatitis E, typhoid, diseases we pick from dirty water or dirty food. For you to have that disease, you're going to assume somebody contaminated the environment. Then you can make other underlying assumptions depending on the biology of what you're modeling. You're going to assume when somebody goes to the environment, the pathogens will remain there before you go and pick up the infection. So I want you to just refine this, and then you can share with what you have, with the assumptions that you have made. It's the same for vector-borne diseases. We know that some diseases, I know you looked at this yesterday, some diseases are indirectly transmitted. For example, malaria is transmitted by the mosquito. But you can refine this and put exposed class here. You can also expose the mosquito because it doesn't get infected immediately upon biting. It's going to take some time. If you look at, when you get time, you just go to Google and look at the life cycle of malaria in the mosquito. You will see that it is a long process. But here we have ignored E. And the assumption is that maybe the importance of this model wasn't to look at that stage in the mosquito. But it takes a long time before, when the mosquito picks the female and male gametocytes, when it bites you and you have malaria. Those two have to fertilize, come to become cysts, then larva, then pupa, then what, before they come out in the mosquito gut to bite you again as now a sporozoid. So when you look at this, you see a lot of assumptions have been made to simplify that long process into this simple process. Even here, when we are bitten, the parasites go into the liver, they spend there 2 to 3 days, then they come to the blood, they attach themselves to the red blood cell. Between 2 to 3 days, the red cells start rupturing, then they again releases more parasites into your body. That's why when you have malaria, you feel bad or you feel cold at the same time of the day. The red blood cells have ruptured and now they're attacking your red blood cells. You even start shaking. And if you're in the village, they will have a mat over you. I don't know where the psychology is, and for some reasons it works, but the parasites have, the red blood cells have ruptured and they're attacking the new red blood cells. So as you have that attack at the same time with so many red blood cells being destroyed, you feel cold, you start shaking. And if the process continues, you'll become anemic and die. But here we have only included eye. Those details have not been included in this model. And that is because they have made some assumptions. Maybe we want to see how malaria is at population level. We just want to see what happens at population, not inside the person. So you look at a model, you should be able to make an assumption, or to read the model and know the type of assumptions that were made. So in summary, if you're modeling a system where you have absence of recovery, you're going to have a logistic epidemic. Or if you have a logistic epidemic, then you know there is no recovery. And what are some of the cases of such diseases, like HIV? People continuously become infected until the population is depleted, right? So if that is the case, then you know that you have, or you expect, a logistic curve. As you model or as you fit, it must be a logistic curve. Now, if you don't have susceptible recruitment, that is, when we say no susceptible recruitment, you should be able to know by now. It means there is no natural birth and there is no loss of immunity. So you're going to have your SIR. People stop at R. So when you just read there, no susceptible recruitment, that means there are no demographics. There is no loss of immunity. People are permanently recovered. Then for simple epidemics, an epidemic is a disease outbreak. They are always short, like for a period of a year or so, like we had cholera. They are going to be short and different from the endemic, which persists in the population, like malaria and HIV. So when you have simple epidemics, you can assume no recruitment or no demographics, no natural death, no birth. Then you assume there is no loss of immunity. People remain completely recovered. So when we look at susceptible recruitment through birth or loss of immunity, then we have recurrent epidemics. This means that the disease is recurrent within the population and it remains there for life. So that means that when you have a recurring epidemic like malaria and HIV, you cannot really model without, you can't model without the demographics. Why? Because this disease is here for a long time, and we need to incorporate the birth, death, loss of immunity, and so on. So when you're making assumptions, you should always study the disease first. What are your objectives? What is the disease you're modeling? What is your objective? All those can lead you to the assumptions that you're going to have to make without losing meaningful biological concepts of your model. So in summary on assumptions, apparently assumption is believed to provide an easy or lazy person's way out of what seems to be, at least at first glance, a perplexing situation. A lazy, or it provides an easy way out. At least one is not just justified in going ahead and making some assumption, which moderately expressed is wholly an arbitrary one. Accuracy of interpretation never can be supplanted by assumption. For example, we can be looking at HIV. I assume one thing, you assume something else. And the other fellow can also assume different things from each of the different from others. So I saw that we are 15 on the Zoom. That means we have 15 different assumptions independent of each other. So grant the acceptance of assumption, and then any one of the 100 different solutions is correct. With all the assumptions that we have made, if they are all acceptable, we are going to have 15 different solutions that are correct. So although they say that an assumption is as good as your result, or your output is as good as your assumption, you have to be very, very keen, you have to be very careful, and you have to know the assumptions that you make. We can actually try to do that and look at the different. We can look at the just one model. Then each one of us can make an assumption. Then you will see how different our results can be. Now, I don't know about the time, but I think we have a break. Is it at 10:50? Hello?
We have a break at 10:15.
To 10:30?
Yeah, to 10:30.
Okay. So maybe we can have the break now.
Okay. And
Then we
we
can,
come back at 10:15.
yes, if we have time. No, it is almost, yeah, it is 15 minutes. I think we can choose one model. Let's first agree to which model. Can we just pick one? We make different assumptions, then we see how assumptions are important in our models.
Professor Chivumbi,
Yes.
first of all, thank you so much for this presentation. We have a number of models we have built ourselves, and the purpose of this was to refine some of our work such that we could make it strong and be liable for submission. In yesterday's discussion, we realized that actually we would improve a lot, and actually we now understood why in the other journal club you were querying the vaccination, the malaria vaccination in the Bugiri model. Although at that time we didn't understand it quite well, but in yesterday's session, we understood why. And actually, even in this presentation, when you go to slide number 12, you've actually hinted on what we discussed yesterday on vector inclusion. So my suggestion is that we could have, for example, Brian Mushave or Maureen share their, Maureen, if you have the dengue model, or Brian, if you have the active. Brian, did you work on active TB or latent TB?
We did latent TB, but Jessica is the one with the work. Let me get in touch with her, maybe she can share.
She's not on the call?
Then
Let's just get somebody who is here, such that we don't waste time, because you could just share your screen. Yeah, Jessica is here. We could, Jessica, can you hear us?
Yes, I can hear you.
Yeah, let's share the assumption is let's share our model and the assumption is we made such that we can improve them.
Okay.
I think the best way we can do this, Abe, is that they can share their model.
Okay.
And then I'm going to tell them the assumptions they made before looking at your assumptions, by just looking at their model.
Very good.
Then we can compare.
Very good. Thank you so much, Prof.
Can we do that now, or do you want us to do it after the break?
Just now. We are still strong, Prof.
Okay, then you share. Then I'm going to tell the assumptions you made in your model.